Hi, everyone. My name is Dr. Tess Eidem. I'm a microbiologist and environmental engineer researcher who helps cultivators and manufacturers overcome microbial challenges and build good practices in their processes. And today we're going to be talking about microbial players in indoor cultivation, benefits, risks, and remedies. So cannabis is kind of actually in a very innovative space. It's really predominantly grown in these indoor cultivation sites, which is called controlled environmental agriculture, or CEA. And lots of high value crops are actually brought indoors to kind of help protect them from these environmental impacts like hail and flooding and things like that, pests. And bringing these high value crops inside can really increase your yields. It can increase the quality of products. It also helps build local economies because you're not bringing in, stuff from California, but you're growing it right there, and it's fresh. And it's also thought to be a new way to build more sustainable agriculture or horticulture in these indoor CEA environments. And CEA is still a very ever-evolving industry, but it also still has its own unique challenges for these indoor agriculture cultivators. And like I said, cannabis is really specifically grown in these indoor environments, also a little bit in greenhouses and outdoor, but most smokable cannabis is actually grown in these indoor CEA environments. Cannabis is a little bit different from our leafy greens and sprouts and culinary and medicinal mushrooms and tomatoes because it's both a medical product and a recreational product. And it's also not only orally consumed like most of these fruits and vegetables are, but it's inhaled. And so in reality, because of the medicinal nature and the inhalation route of administration, we should be growing cannabis in a very controlled, high quality environment. But because cannabis is still federally illegal, there actually aren't any federally established quality and safety standards for cannabis cultivation. And to kind of compensate for this, cannabis products are, instead of, going through very clean environments and making sure that the indoor cultivators have all their sanitation records and HACCP plans and things that other, indoor cultivation or indoor manufacturing has to do. Instead, what the cannabis industry really relies on is rigorous testing of that cannabis in its final product form. So on this next slide, you're going to see all the different types of tests that are performed on these cannabis final products from third-party testing labs. And it really varies depending on the state that you're in. But you'll test for the active pharmaceutical ingredient, API, your THC, CBD, and the acid forms. You're going to be looking at the potency. Also, for quality aspects, you might want to know what your terpene contents are. And then there's a lot of contaminant testing, like for looking for pesticides, residual solvents, heavy metals. Making sure that your product is not too moist. So looking at the water activity or the moisture content of those final products, especially in flour or food products. You want to make sure if you're storing this on the shelf that it's not going to allow for microbes to come back and grow. And then foreign material inspection is another way of looking at the quality of that end product. But a large amount of testing is actually performed to look for microbial contamination, either by specifically looking for microbes up here in the top right, doing qPCR, so that's quantitative polymerase chain reaction. So that's looking for the unique fingerprint of the DNA of certain microorganisms. Or if we're looking at these indicator microorganisms, which are these broad groups of organisms that you want to count, and you set a threshold and you say, basically, above this threshold, these microbes have too high of a bioburden and there's something wrong with this product. It either spoiled or wasn't grown in clean conditions or it wasn't handled properly. And so you have these different levels of contaminant testing, active ingredient testing, and just overall quality testing that's performed by these third-party testing labs. But there's lots of ways to get around these testing mechanisms that we have in place in cannabis currently. And many folks have probably heard about lab shopping. That's specifically for potency, but it can also happen to try to overcome these different microbial failures. So folks might only treat their samples that are going out for third-party testing, but not treat the rest of the batch, or they might shop around until they get the results that they want on the microbial levels, or if there's contaminants there. And so even though it gives us a sense of safety, and it's very important to do this third-party testing, it doesn't actually get at the heart of why these products may have contaminants on them. And it doesn't really tell you how to grow it, how to manufacture it, how to handle it. It's just looking at that snapshot in time. Did the product pass or not? If it did, it can be sold. If it didn't, there may be ways to recover it, but otherwise you might have to dispose of that product. And really zooming in on the microbial contamination factors that are really tested for in cannabis, we have to first zoom way back out and look at the tree of life. So this is a phylogenetic tree. All life on Earth stemmed from this very bottom green box here at the base of that tree. And that's our last universal common ancestor, or LUCA. And in cannabis, we don't look at all microbes that are on the plant. We kind of hone in on a few groups of microbes and a few specific pathogens. So if you're looking at this tree, you'll see eubacteria, or true bacteria, are here now outlined in purple. There's lots of different microbes that are on this entire branch of the tree of life. And in cannabis, we look at biotolerant gram-negative bacteria, Enterobacteriaceae, coliforms, and total aerobic bacterial count. So some of these microbes will fit into multiple of these family groups. Some will be unique, and then some microbes don't even fit into the ones that we test for. And these broad groups really do include, you know, hundreds of thousands, if not millions, of different microorganisms, but we really want to focus in on those that we know are linked to quality, or we know are linked to good sanitation. For example, coliforms are pretty much exclusively found in the gastrointestinal tract of animals. And so if you find coliforms on your product, it likely came from fecal matter or sourced in that way somehow. And then we'll specifically look at some of these microbes like pathogenic E. coli and Salmonella by qPCR. Now, when we look a little bit further up the tree of life, the next microorganism that's group of microorganisms that's tested for are fungi. And it's always important to know these microbes because fungi is actually more closely related to plants and animals than they are to bacteria. So that's really, really important when thinking about and understanding fungi compared to bacteria and the unique problems and environments and challenges that they pose. And a lot of times fungi are sort of binned into this total yeast and mold category, or TYM. You may know like colony forming units, or CFU per gram. You will enumerate the amount of total yeast and mold on your product. And then if it's below some threshold, you'll pass. And if it's above some threshold, then you'll fail. Now, in this TYM category, there are some species that are tested for specifically, like Aspergillus species. Now, Aspergillus is a genus of fungi, of mold specifically, that includes about 250 different species, and only 4 are tested for in cannabis. And those 4 species are associated with lung disease, either actively infecting somebody and causing disease that can transfer to the brain, things like that, like aspergillosis, or they produce different toxins. And actually, a lot of molds and fungi produce toxins, and we only look for a handful of them. And these microbes, again, it's really important to understand the different microbes that may be playing a role in your cultivation environment. Because, I mean, number one, because of compliance risks. So 10 to 30% of flower products actually fail due to microbial contamination. And that's a pretty conservative estimate. Most people who fail don't particularly like to talk about it. So we do think that it's pretty underreported. And when it comes to recalls, microbial contamination really dominates the reason why these cannabis products are recalled. And these microbes, they fall into lots of different categories. They can be plant pathogens, human pathogens, post-harvest spoilage microorganisms, and they can also produce all sorts of byproducts that can be harmful. So some of these microbes that are pathogens will actively infect and cause disease in plants. Here's a picture in the middle of powdery mildew, and that can transfer to other healthy plants and really decrease yields, decrease plant growth, things like that. Also, it can be foreign matter on your final product. Human pathogens can spread in a facility. They can either some plant pathogens can infect humans, like Pseudomonas aeruginosa. Even some fusarium species can infect humans. And so there may be some pathogens that can, spread the gambit between plants and humans. And some are post-harvest spoilage microbes. They may be around in your garden at low levels, but then they can flare up if given the right conditions. Like if you don't dry cure in a drier, less humid, kind of cooler environment, you may allow these microbes to sort of rear their ugly heads and spoil your product. And then one thing we don't really talk about that much in cannabis are the byproducts of these microorganisms, which can be a risk to both consumers and to workers. And these are the mycotoxins that we talk about. There's also toxins that bacteria can produce that are known immune activators that we don't test for in cannabis, and allergens, which we actually don't test for in cannabis, but many, many molds produce allergens that are problematic, not only for cannabis cultivators, but for other built environments, other indoor environments where humans spend 95% of their time. They interact with these airborne allergens and can have all sorts of negative health consequences because of that, from asthma to other chronic lung diseases. And so it's really important to understand the risks that each of these types of pathogens pose, or even microbes or their byproducts pose, not only to compliance, which is what we normally think about in the cannabis space, but also to your end user, since we're inhaling these products and it's known that these microbes don't die because of combustion or because of vaporization. They actually can survive that process and make it into your lungs. And so can their byproducts like allergens and toxins. And so you want to protect your workers too, who are trimming this potentially moldy weed and breathing that in. That can also cause negative health consequences for workers. So on the end, all in all, we want these microbial risks to be taken seriously, but it's also important to mention that not all microbes are bad. In fact, most of them are pretty good or neutral in your garden. On the left here is a figure that shows different beneficial microorganisms that are used as biocontrol agents for botrytis. Microbes can be used to control insects. They help with nutrient uptake. They help compete against bad microbes. Just the fact that they're there and they're competing for the same ecological niche can help root out and get rid of some of the pathogenic microbes that could be in that environment. And then there's all sorts of plant microbe synergies that have a role that we still don't fully understand. And scientists are still investigating. And one that I thought I would share because I thought it was really cool is this very recent paper, just came out about a month ago. talking about the Pseudomonas species, which are bacteria. And some Pseudomonas species are actually pathogens, some are not, some are beneficials. But this particular group of Pseudomonas species were friends to plants, and they produce these antimicrobial peptides that actually were antifungal against plant pathogens. And they kicked so much butt against botrytis that the researchers who identified these little antimicrobial peptides named them after Keanu Reeves because he also kicks a lot of butt, and they named them Keanu because of how effective these molecules are at protecting plants from fungal pathogens. And it also protects the little Pseudomonas bacteria from amoeba trying to eat those bacteria. So as scientists, we're still, elucidating and uncovering how these microbes really interact with and can benefit and can harm plants. And it's a constant research process. And that's also the mentality that the indoor cultivator should adopt, as well as they're trying to understand their indoor microbial ecosystem that they're creating for different microbial species. So really trying to get an understanding of the microbes that live in these indoor environments means that you've got to look at your entire process. So here on the right, it's working through that cultivation, dry cure, packaging process. And then knowing where each of these microbes are really coming into that process. Some microbes are just going to be in the environment, in the indoor environment. Aspergillus lives in indoor buildings. It can grow on drywall. It can grow in all sorts of areas without plants being there, but then introducing the plant matter and high humidity and other substrates like raw materials. Here is the second bullet point down. Your cocoa, your other materials that you're bringing into your facility may also be harboring microorganisms, some of which are going to probably be fine, but some may include pathogens and other microbes that you don't want in your garden. I've tested cocoa and I've seen lots of different microbes that you don't want, like Aspergillus and E. coli in those raw materials. So really understanding what you're bringing into your facility through your raw materials, and then also what are you allowing to thrive? Water quality is another big one. You may test your municipal water or have your well tested every year, but what about the water that's actually getting to your plants? So it's been sitting in nutrient tanks, it's been flowing through your drip lines. Now what do those microbial populations look like on the other end? And I've definitely been to grow houses where you cut a drip line in half and you can see a microbial biofilm, which is just an organized group of bacteria and fungi that really are coating the inside of that drip line. So it's important as a cultivator to know what those microbes are and to make sure that you're not enriching for the bad microbes that are there. Some microbes are just going to naturally live on your plant. They may not cause harm. Some may be pathogens. They may cause disease, and they may be brought in from getting new clones from a supplier, or they could be brought in from humans. So bringing in, if somebody works in another grow or they have another job and they're they're working in animal husbandry or something like that, and they bring in microbes from other environments, or they introduce their own human microbes. If they're sick and they introduce E. coli or something to the product, then that's another way that microbes can get into your garden. And then spoilage bacteria, we talked about this, and mold, they're usually around at some baseline level, and they can flare up if given the right conditions. And an important thing to note is that while not all microbes are bad, when you're intentionally introducing beneficial microorganisms into your garden, it's important to trust your supplier and to test that beneficial slurry of microorganisms. There was a publication that came out just a few months ago testing commercially available beneficial microbe like concoctions. And basically what they found was that a good chunk of those contained a lot of other microbes that weren't listed on the label, which means that those are contaminants. Some actually had plant pathogens in them, and there were some that didn't even contain the microorganisms that they said they contained. And so really understanding these microbes and when to apply them, I've had folks use beneficial bacillus microbes on their plants, but you want to make sure you're spraying that on the soil and not on the leaves or on the flower of your plant, because those are aerobic bacteria and they may trigger aerobic bacteria failures. So understanding all the potential sources where these microbes could be coming in across your entire process is really that first step. Because the truth is that these relationships between cannabis and the microbes that live on it, so on the leaves, on the flowers, that's the phyllosphere, which you can see in this figure here on the left. The endosphere are the microbes that live inside the plant in the vasculature of the plant. And the rhizosphere, which are microbes that live in association with the roots of the plant. Understanding those microbes is really, really complex, and what they're doing at different times throughout the process, how they can be helpful, how they can be harmful, is just starting to be elucidated by scientists. Both of these publications in this slide here are less than 3 years old. So we are still understanding from a scientific perspective how these microbes play a role and where they are and what the spatiotemporal relationship is between these microbes and parts of the plants and parts of the process. So it is complicated, and that's okay, because there's a good place for cultivators to start. And that's by performing something called a root cause analysis. So when you're failing and you're relying heavily on remediation to recover this failed product, that's really not a long-term solution. And in the long run, it's going to reduce the quality of your product. It's, having your plants be infected with disease or being spoiled in post-harvest processes is going to really be detrimental to your brand, to your company, and it's going to potentially lead to more recalls. So the best way you can address these is by understanding where these microorganisms are coming from. So the first question you ask yourself is, what organism am I actually failing for? If you keep failing for total yeast and mold, see if you can get more information about those microbes that you're failing for. Typically, you can work with your third-party testing lab to ask them to culture or identify the top 2 to 3 microbes that they have been finding on your final product. If they can't do it, they can send it out for sequencing. It might cost a couple $100, but then you know what microbes are your problem microbes so that you can trace them back to the source. Where could that microorganism have been introduced? So, for example, biotolerant gram-negative bacteria, they typically need more water than a lot of total yeast and mold organisms. And so they usually like to live in these aqueous environments. They'll be in your drip lines, they'll be in your soils. They tend to live inside the plant. And so they're more endophytic. So if you know that about your microorganisms that you're failing for, you might have an easier time tracing them back. And if you know the specific species, then you can start testing for and looking for that species up your entire process assembly line. And then how is the microorganism thriving? Like I was saying, BTGN, they like water. So if you're giving them water and nutrients, everything that they need, and you're never cleaning out your nute tanks or your drip lines, that's how they're thriving. They're establishing themselves, they're establishing their little ecosystem right there, and they're going to continue to thrive until you put in some preventive measures. And that's that next bullet point. What preventive measures can be put in place? What can I do proactively to address these microbes? Start flushing your drip lines, start cleaning your nutrient tanks. And then how do you measure efficacy? Once you've done that, how do you know that that's working? Or how do you space out flushing your drip lines? Do you do it every month? How do you know you do it every month? Oh, because you're testing every month and you know that if you clean every month, these microbes don't come back. But if you put it off every 3 months, you're going to see more of those microorganisms that are problematic for you. And you can use this for other problems like aspergillus. Aspergillus is found in many different substrates, and it can also spread through the air. So understanding where these microorganisms are coming from, where they're being sourced into your facility, and then how they're spreading, putting in the preventive measures, and then making sure that you are actually measuring and verifying that your efforts are working is how you really root out these microbial problems. And there's a lot of different good practices that we can take from other industries, in addition to the root cause analysis, that you can integrate into your process. So here, we've got a lot on this slide. It's really intended to show you there's lots of ways to manage your microbes, and we don't have to reinvent the wheel. These practices have been established in other industries, and we can really adapt them to our needs. So good sanitation practices is number one. On the left here is a table that goes through all the different potential sources to address contamination, whether it's from your planting materials like your substrates to controlling plant or human introduced pathogens. There's measures that you can put in place. This is well established in the literature or in other agricultural industries. How can you proactively address those? Quarantine protocols. That's another thing that I think really has a huge impact for a very little input of energy and resources. If you bring in new clones or new seeds to your grow, quarantine those plants until you can test them and ensure that they're not going to be bringing in pathogens to your facility. Same with when you start seeing diseased plants, you need to quarantine those plants and get them away from your healthy plants. Otherwise, they're going to spread disease. Another thing that's really important is a process called hygienic zoning. And you can see this diagram up here on the right, the top right. It's a little building outline of different rooms within a manufacturing site. And they'll have some areas that are designated as high hygiene areas. This is where your cleanest processes are taking place, usually closest to final product, but also you want your mom room to be pretty high hygiene as well. And then you can also see that there's different traffic and worker flow through these areas, with the idea that you should always start in the cleanest areas and work your way to the dirtiest areas. So that helps you to start prioritizing your activities for the day. If you have to fill pots, you should probably do that away from anything that's final product, away from your moms, and towards the end of the day. So you're not, bringing any potential pathogens from that plant substrate out into the garden with you and spreading it around. Environmental monitoring is another really good way to get eyes on your microbes. You cannot see microbes with your naked eye until they're in the millions of CFU. And by that point, you already have spoiled your product, or your plant is already infected. So how do you get eyes on your microbes and know when they're reaching some threshold right before they become a problem? And there's lots of tools out there to get eyes on your microbes, from your traditional light microscope to active air sampling mechanisms to particle counters to Hygiena ATP meters, which you can use to test surfaces and water. Plates, agar plates, there's lots of different media out there that you can use to cultivate all sorts of microorganisms from all sorts of sources. And then there's lots of antigen tests out there, kind of like a COVID test. It can confirm that you have a specific plant pathogen, for example, so you know what microbes you're tangoing with and you're not left guessing. And then finally, a hazard analysis, which kind of the old school way of calling this is a HACCP, or hazard analysis across critical control points. The hazard analysis is really taking a huge step back bringing in multiple members of your team across all the processes that you carry out in your facility, from good agricultural practices on the left to good manufacturing practices on the right, and really assessing the biological risks that you could be introducing along your entire process. So in cultivation, we know water, raw materials, air quality, environmental controls. If it's really humid, you're not circulating that air, you're going to probably have mold problems. You're creating an environment where those microbes can thrive. So where are you going to put in preventive measures or really start putting in environmental monitoring efforts to get eyes on these microbes and be proactive in addressing these biological risks that are going to come into play along your entire process? And these hazard analyses are so useful for cultivators. And once you start thinking this way, it becomes much more intuitive and much easier to be proactive in your microbial risk management. So, in summary, cannabis really is uniquely poised as a CEA trailblazer, not only because it's a medicinal product and a consumable good, but also the quality should just be higher. We're inhaling it. It's for medical patients. And so we really should be taking the reins to adapt some of these good quality practices from other industries into cannabis. However, it has been a challenge for the industry because there's really no enforcement that we are growing clean and producing clean product. And so it's a tough place to be in for cannabis cultivators, because everyone wants to protect consumers and workers, but we don't really have these practices established in our industry. And we also need to kind of reframe microbes a little bit in cannabis and recognize that not all microbes are bad, but it's about getting eyes on the microbial landscape in your facility. Which microbes are where and when are they going to be problematic, so you can be proactive in addressing them ahead of time before they, take out an entire harvest. And finally, we can pull these good practices from other industries, perform this hazard analysis, and implement environmental monitoring just to get started. Those are going to be a huge return on investment. It does not cost that much money to start getting basic tools in the hands of cultivators, integrate this into your IPM programs, and really give it legs, give it people to really push these new good practices forward, because it will return on your investment. And with that, I'm very happy to be here today at the Lab Roots Cannabis Science Day. And you can go onto my website. There's lots of good resources there for good agricultural practices, manufacturing practices. And you can always reach out to me by email at Tess at roguemicrollc.com. Thank you so much for attending.